Good morning, everyone. Welcome to the online classes of biology for class 12th. Today we will take up chapter 9, Strategies for Enhancement in Food Production, part 3. The subtopics for today are biofortification, SCP, that is single cell proteins, tissue culture, and somatic hybridization. Learning objectives for this session are biofortification, single cell proteins, tissue culture, somatic hybridization. That means after completion of the session, you will get a better understanding of all these mentioned subtopics and learning objectives. The first subtopic is biofortification. It is the process of breeding of crops to produce varieties with higher levels of nutrients like vitamins and minerals, or higher protein or healthier fats. Some examples are iron fortified variety of rice as golden rice, lysine and tryptophan rich varieties of maize, vitamin A enriched variety of carrots, spinach and pumpkin. Atlas 66, having a high protein content, has been used as a donor for improving cultivated wheat. IARI, Indian Agricultural Research Institute, New Delhi, have released many varieties of vegetable crops rich in vitamins and minerals like vitamin A enriched carrot, spinach, pumpkin, and vitamin C enriched bitter gourd, bathua, mustard, iron, and calcium enriched spinach and bathua. Even protein enriched beans, that is broad beans, lub, French, and garden peas. All these basically are to remove the problem or to combat the problem of hidden hunger. Now about the single cell proteins, also called as SCP. These are the alternate source of the regular food production. Microbes are grown on an industrial scale and processed and used as nutrient-rich food. Example, spirulina. Now, what are the advantages for using these alternate food resources? It is rich in protein and low in fat, can be easily grown on cheaper materials. The use of waste materials reduces pollution and they reduces the pressure on agriculture also. Microbes like spirulina can be grown easily on materials like wastewater from potato processing plants, having starch, molasses, animal manure, and even sewage to produce large quantities, and can serve as food rich in proteins, minerals, fats, carbohydrates, and vitamins. Methylophilus methylotrophus bacteria has high rate of biomass production and growth. It can be expected to produce 25 tons of protein by 250 grams of this microorganism. That means its protein content is very high compared to a crop plant rich in protein. Now, this image is showing the spirulina alga, the powder form and the tablets. These are generally used to get a supplement for protein. Now, tissue culture, also called as micropropagation, it is a technique of generation of whole plant from a cell or tissue of a plant on a suitable medium. Conditions should be aseptic, that is contamination free. Plants produced are genetically identical and constitute somaclones. Advantages of using this process are: it is a very fast process. Many plants can be grown readily. Healthy, disease-free plants can be grown by meristem culture. That means if we are having a diseased plant and we want to get a disease-free plant from it, we can use tissue culture technique by growing the meristem of the plant, that is the apical or the axillary meristem, so we can get a disease-free plant even from a diseased plant. Now, the image is showing the technique involved. The tissue sample scraped from the parent plant. The parent plant is having the desired trait. Now, the part that we taken out from the plant is termed as explant. The tissue sample placed in the agar growth medium containing the nutrients and plant hormones. Later on, the sample developed into tiny plantlets. These tiny plantlets are transferred into the compost to get many plants from it, or we can go for another method in which we can grow these plantlets into a soil-free, nutritive solution containing medium. The capacity to generate whole plant from any cell or explant is called totipotency. Thousands of plants can be produced from explants in short interval of time using suitable nutrient medium, aseptic conditions and use of phytohormones. This method of producing thousands of plants is called micropropagation. Each of these plants will be genetically identical to the original plant from which they were grown. That is, they are somaclones. Many important food plants can be grown by using this technique, like tomato, banana, apple, etc. Now about the meristem culture. The recovery of healthy plants from diseased plants can be done by meristem culture. Although the plant is infected with the virus, the meristem, the apical and axillary is free of virus. Hence, one can remove the meristem and grow it in vitro to obtain virus-free plants. Now, the last topic is somatic hybridization. Isolation of single cells from their plants and after digesting their cell wall, fusing the cytoplasms of two different varieties is called somatic hybridization. and these hybrids are called somatic hybrids. It is the process of fusing protoplasts of somatic cells. Protoplasts are the protoplasm bounded by the cell membrane only after removal of the cell wall. The protoplasts are derived from two different varieties or species of plants on a suitable nutrient culture medium. The steps involved in the process are isolation of protoplast by digesting the cell wall by the use of the enzyme cellulases and pectinases. Fusion of protoplast. It can be done by the physical method by providing high voltage frequent current, so the protoplast get fused, or a chemical method can also be employed, that is the use of PEG, polyethylene glycol. Growth of fused protoplast on suitable medium to form somatic hybrid. Now this is the method. The cells are isolated, then protoplasts are fused, somatic hybrid cells are obtained, and then these are carried forward for the tissue culture technique, and small plantlets can be obtained out of it. After completion of the session, I am sure you all have achieved the learning objectives of this session. Take care.